Thank you for the introduction. So, as you can probably guess, I consider myself to be a synthetic organic chemist. But unlike other synthetic chemists at Rice, who make anything from nanomachines to new materials like carbon nanotubes to surprisingly graphene on toast, I get right to the fundamentals and I make molecules, specifically new molecules. And I think it's one of the hallmarks of the Rice chemistry department, not the entire university itself, that we're known for the discovery of C60, or the buckyball, which won the Nobel Prize in Chemistry in 1996. Now, as a synthetic organic chemist, I think it's absolutely awe-inspiring to think that I can get to be the first person in the history of the universe to be able to connect these atoms in a certain way and see that new creation in a flask for the very first time. And so just to illustrate this, take a look at some of the new molecules that our lab, including myself, has synthesized over the past year. Now, of course, as someone who's interested in organic methodology, I do have to take a step back from this rather gratifying perspective and consider a more relevant question. And that is, how can I synthesize these molecules in a more efficient and atom economical way? And so this brings me to my research project, which concerns a family of compounds known as biochemistry. And so these are part of a larger class of compounds known as amines, and you guys in the audience may be familiar with amines as those organic functional groups that contain one or more nitrogen atoms and are analogs of the compound ammonia. So as it turns out, these bioarylamine motifs are commonly found in a lot of drug molecules and a lot of pharmaceutical products, and you may or may not recognize these drugs, but you can certainly appreciate the range in terms of its therapeutic effects. And so, in terms of the synthesis, it's kind of surprising to consider that despite the prevalence of these types of compounds in the pharmacopoeia, what's surprising is that we have a limited number of ways to make them. And in fact, most of the commercially accessible methods to synthesize these bioarylamines depend on the use of transition metal catalysts and ligands, and often harsh reaction conditions like high temperatures and strong oxidants. And so these types of reactions are perfectly fine to run in the lab, but you'll quickly run into some problems once you consider a more large-scale or a real-world application, for example, in the pharmaceutical industry. And so the goal of our research project is to develop a mild, but more importantly, a transition metal-free method to synthesize these bioarylamines, which is quite desirable from both a practical and environmental standpoint. And so to put a long story short, our lab successfully devised such a synthesis, and one of the highlights of our method was to use a polarity reversal strategy. It's really cool because I can turn the nitrogen atom from being partially negative, which is what it is most of the time, to being partially positive, which means it can now accept electrons and form that critical carbon-nitrogen bond. And for that, we developed a whole new family of reagents, of polarity reversal reagents, to be able to do that chemistry. As you can see from the picture, the compound, or the reagent, is a beautiful white crystalline solid. It's super easy to make and super easy to use and absolutely bench stable. And in fact, we just recently patented this reagent, so laboratories around the world will soon be able to purchase this reagent and use it for their own chemistries. And in fact, the idea of polarity reversal opens up a whole new avenue for investigating new kinds of reactivity. And so I actually want to go back to one of the first slides to mention that these molecules are not arbitrarily chosen. In fact, they were all made using that same type of reagent that I just described. And so there's massive applicability and versatility to this method. And so you can not only make these bioarylamines, which I just mentioned, but you can also make these 3, 4, and even 5-membered nitrogen-containing rings. You can even make these 7 to 9-membered fused ring systems, which are really difficult to make using other methods. So hopefully you guys in the audience can just appreciate the sheer generality of this method, which I think is one of the strongest points of this work. And so unfortunately, 3 minutes is not a long time to talk about my research project, but hopefully I'm not so much as informed, but rather inspired you with an amazing research opportunity here at Rice. Now, of course, while getting your work published in a reputable scientific journal and getting it featured in the lead story of CNN magazine, some of the great perks of a successful research project, I think one of the main takeaways here is that I get to learn some amazing chemistry from outside the classroom setting and to be able to do that with some amazing people, and to truly understand what it means to be a scientist and an active player in scientific research. Thank you so much, and I'm happy to answer any questions during the reception.